Hello, and welcome to our famous video on plants, their growth, development, and structure. This is going to be a wonderful time. We are going to begin with structure, and I want you to think of plant structure similar to animals. Because of that, because plants are made of different cells and tissues, and all these parts work together like a machine to create, for their one common goal or purpose. And there are two main parts of plants. There are roots and shoots. Okay? Hopefully you got my arm gestures there. Understand what that means. Roots are firmly anchored in the ground, retrieving nutrients for the plants and spreading them throughout the entire plant. In eudicots, there is a major root called a taproot, which is similar to like a carrot. A carrot is a taproot, for example. And off of the taproot, there are many lateral roots that come off of them, and they help in the transferring nutrients. But in monocots, there is no taproot. Also have root hairs, which increase the surface area, which you can think of as like microvilli in your intestines. So that's pretty sweet. Shoots, they shoot up from the ground. You can think like a rocket ship shooting up. And shoots consist of a system of stems and leaves. And these stems are used to divide and raise the leaves. Stems have nodes and internodes. Nodes are where the leaf is attached to the stem, and the internode is the part in between. They also have axillary buds, which form at the node and can develop into a lateral shoot, much like a lateral root would. An apical bud is a bud located at a tip of the plant that grows outward and has more photosynthetic activity because it is the highest of all the points. And you can think of that like it's apex. And this is considered apical dominance because it has, like I said, an advantage over the other buds. Leaves are the main area for photosynthesis to occur, and most leaves are considered blades. Leaves also have petioles, which is where the leaf connects to the node. And plants have major tissue systems, which are three types, which are dermal, ground, and vascular. I repeat, dermal, ground, as in the ground, and vascular. Okay. You can think of dermal like a dermatologist, like skin, so that's the outer layer of the plant, like the skin of the plant. There is the ground tissue, which I like to think of as kind of a misfit, because ground tissue contains cortex and pith is internal in the vascular system and is present in most stems, but only in some roots, and cortex is internal to the dermal tissue. So it's in between the dermal tissue and the vascular tissue. And then the vascular tissue system has two subtypes of tissue, xylem and phloem. Xylem I consider like the beverage bro, the guy that carries the water and spreads it throughout the plant. And phloem is the food guy. Food, you get it, like PHOOD, food. And he carries the nutrients throughout the plant. Phloem and xylem together are known as the stele. And this concludes our section on structure. We will now move on to growth.
Plants. You would think that they're very alike to humans. However, plants have one major difference between humans and other animals. Plants grow indefinitely through all of their lifetime. This is called indeterminate growth, because at no point will the growth cease. Many other animals and certain structures of the plant only grow to a certain point. This is predictably called determinate growth. The reasons that these plants can continue to grow in this fashion is because of undifferentiated tissue called meristems. There are two different types of growth for this plant: primary growth and secondary growth. Primary growth is when the plant grows from the extremes of its exterior. For example, if this plant were to grow outward this way, that would be primary growth. There's also another type of growth called secondary growth. Secondary growth is when the plant grows in thickness. If this branch were to expand, that would be secondary growth. Primary growth uses apical meristems, while secondary growth uses lateral meristems. The materials that can grow in primary growth are vascular cambium, vascular tissue, and cork cambium. When the conditions are ideal, the cells of the meristream divide quicker than usual, and that's what allows growth. The various plants of the world have different life cycles. Annual plants germinate, flower, seed, and die all in 1 year. An example of this would be a wildflower that only lives for 1 year. Biennial plants do the same, but fruit only in their second year. For example, one year they would flower and then another year they would fruit. Perennials complete their cycles over many different years. This is what we usually see in bushes and trees, plants that live for many years. Now we'll move on to how a root grows. Root's tip contains the root cap, which main function is to guard the apicable meristream from any sort of damage. There are different zones where the cells function. The zone of cell division is where new roots are produced. The zone of elongation is where the root grows in length, and the zone of differentiation is where the cell becomes the different types of various tissues. The endodermis is the thick inner layer of the cortex. Now to shoot to the growth of leaves. Leaves are created by the leaf primordiae, which are thin extensions of the apicable meristream. Now we'll go back to the epidermis. The epidermis is covered in little pores called the stomata, allowing exchange of gases within the plant. These pores are regulated by guard cells, which allow some gases to escape or get in. The leaf's ground composition is called mesophyll. The mesophyll has two different layers. The palisade mesophyll contains the longer parenchyma cells, while the spongy mesophyll has loosely arranged parenchyma. The leaf continues to grow as the vascular tissue of the stem grows. In addition to primary growth, the roots and shoots of plants can grow thicker as well, classifying as secondary growth. The vascular cambium continues to grow as the meristhecum cells divide, increasing the diameter. Now you know how cells in the shoots and leaves grow.
The specific series of changes by which cells form tissues, organs, and organisms is called development. These changes are controlled by environmental factors and genetic information. They also have the ability to alter form in response to local environmental conditions, which is called developmental plasticity. This is like when a plant's leaf shape is altered whether or not they are submerged in water. There are three overlapping processes in development: growth, morphogenesis, and cell differentiation. Growth is an irreversible increase in size. Morphogenesis is the process that gives a tissue, organ, or organism its shape and tells where each type of cell goes. Differentiation is the process by which cells change into each type that are needed in the plant. During cell division, new cell walls develop from the cell plate during cytokinesis. The exact plane develops during interphase. Microtubules become concentrated into rings called the prophase band, which goes away during metaphase, but tells the future of planes of cell division. The plane does not determine the shape of organs, but the symmetry, how the cytoplasm is distributed during cell division, determines cell fate. Asymmetrical cell division, which is where one cell receives more than the other, usually signals a key event, such as in epidermal cells. The smaller epidermal cell then becomes a guard cell. Asymmetrical cell division also establishes polarity as well, which are structural or chemical differences at each end of the cell, such as in plants, which have an axis which determines a root end and a shoot end. These can be found in physiological differences as well as morphological. The first divisions of plant zygotes are normally asymmetrical. Plant cells expand mainly on the cell axis. Pattern formation is the development of specific structures in specific locations. There are two types of hypotheses about how the fate of plant cells is determined. Lineage-based mechanisms say that cell fate is determined early in development, and position-based mechanisms say that the cell's final position in an emergent organ determines what kind of cell it will become. Developing cells can synthesize different proteins and diverge in structure and function, though they share the same genome. Cell differentiation depends on the control of gene expression. The activation or deactivation depends on cell-to-cell communication. Plants have stages of development, which go from juvenile to adult vegetative to adult reproductive. These are called phases, and the morphological changes that arise from these transitions are called phase changes, which occur in a certain region called the shoot apical meristem. From the juvenile phase to the adult phase, the most obvious morphological changes typically occur in leaf size and shape. Nodes retain juvenile status so that new leaves that emerge from juvenile nodes will be juvenile as well, even though other leaves on the plant may already be adult. A mature plant may be induced to flower. Flower formation occurs during the change from vegetative to reproductive phase. It is cued by environmental cues and internal signals. The production of a flower by a shoot, apical meristem, stops the primary growth of that shoot. The transition from vegetative growth to flowering is associated with the switching on of floral meristem identity genes. The order of each primordium's emergence determines its development into each floral organ. These organs form four whorls. Organ identity genes encode transcription factors that regulate the development of these whorls. The ABC hypothesis of flower formation proposes that three classes of genes direct the formation of the four types of floral organs. A gene is switched on in the two outer whorls, which are sepal and petals. B genes are switched on in the two middle whorls, petals and stamens, and C genes are switched on in the two inner whorls, stamens and carpels.
That concludes our video. Thank you.